All right, well, hello, and welcome to the third and final heat of the University of Manitoba's 10th annual 3MT competition. Today's heat's being recorded and broadcast live via a Zoom link that's available on the Faculty of Graduate Studies website. Before we begin, I would like to start by acknowledging that the University of Manitoba campuses are located on original lands of Anishinaabe, Cree, Oji-Cree, Dakota, and Dene peoples, and on the homeland of the Métis Nation. We respect the treaties that were made on these territories. We acknowledge the harms and mistakes of the past, and we dedicate ourselves to move forward in partnership with Indigenous communities in a spirit of reconciliation and collaboration. My name is Randy Jamieson. I'm an Associate Dean in the Faculty of Graduate Studies, and I'm going to be the MC for the event. 3MT is a research communication competition that challenges students to consolidate their ideas and work so they can be presented concisely to a non-specialist audience. The competition provides career-building opportunities for our students. It celebrates and promotes the research done here at the University of Manitoba, and it builds connections and opportunities for our student researchers to share their work with the city and with the province. We're proud to be associated with an inspiring group of graduate researchers who are here today to share their work with all of you. I'm certain that you'll come away enthused by the work being done by each of these exceptional students and presenters. This year, 72 graduate students from the University of Manitoba submitted a video application to be part of this competition. All of those submissions were reviewed by assessors from different faculties here at the university, and a final group of 35 was selected to compete in the three heats. Of those 35, 11 of the challengers will compete in today's heat. The top three ranked presenters here today will win a $200 prize and will advance to a final competition that will also be held online on April 7th at 7 p.m. And all of you are invited to tune in then to watch that event as well. Our three judges today are Mr. Greg Mackling. Greg is a co-host on the 680 CJOB morning show and the host of the Blue Bombers halftime show during home games. Greg is active in the community with a variety of special projects and events, including his role as spokesperson for the St. Boniface Hospital Foundation Lottery. Dr. Sandra Weber. Sandra is a physiotherapist and an associate professor in the Department of Physical Therapy in the College of Rehabilitation Sciences here at the University of Manitoba. Her research focuses on measuring physical activity, ambulation, and sedentary behavior in older adults and people with chronic disease. She often uses wearable technologies to provide objective measures of what people are doing in their everyday lives. And Dr. Jake Stout. Dr. Stout is an assistant professor in the Department of Biological Sciences here at the University of Manitoba, who specializes in plant biology and genomics. So welcome, judges, and thank you for being here today. To choose who will advance to the final competition amongst all of the presenters that we're about to see. Our judges will be assessing our competitors' presentations on the basis of comprehension, engagement, and communication. They've been provided detailed criteria for their evaluations. Each competitor will have only a single overhead slide and 3 minutes for their presentation. The timer will begin when the presenter begins speaking. So we're going to begin with the first 6 challengers. We're then going to take a short 3-minute break and then finish the competition today with the last 5 challengers. After all of the challengers have completed their presentation, the judges will have 10 minutes to tabulate the results and choose the top 3 challengers who will move on to the final 3MT competition here later on at the University of Manitoba. If you're posting online on social media today about the event, we welcome you to do that and to use the tag for University of Manitoba Faculty of Graduate Studies, which is at Grad Studies on Twitter and at UManitoba on Facebook. So with that, it's time to start the competition. And our first challenger today is Erwin Tigium from the Department of Medical Microbiology and Infectious Diseases. Erwin's thesis title is Detecting Active Tuberculosis in People with HIV. Erwin, you can begin speaking when you're ready, and the timer will start when you do that.
Here in Manitoba, we have one of the highest tuberculosis or TB infection rates, as well as HIV infection rates across Canada. 1 in 3 people who are co-infected with HIV and TB died, making this TB disease the leading killer in HIV-related deaths. But the thing is that both TB and HIV are treatable. However, our current tools to detect TB disease are inaccurate. And if a patient is infected with HIV, it makes it even harder to detect this lung disease, leading to over 50% of people not even aware that they're infected with TB disease. So the goal of my research is to come up with a rapid test that can accurately detect active tuberculosis in people with HIV. So when we get bacterial and viral infections, our immune system produces specific genetic and protein material to fight against these infections. Almost like a fingerprint, each response is unique and distinct to a specific infection. So for my research, I aim to find the best genetic and protein fingerprint that best describes active tuberculosis disease in people living with HIV. To do this, I will be comparing two groups of people. First are people with active TB only, and second are people with active TB living with HIV. And using a method called RNA sequencing, I will scan the genetic materials between these two groups and find the best genetic fingerprint that best describes active TB in people living with HIV. From there, I will conduct an experiment called multiplex assay, where I will measure the protein materials between these two groups and find the best protein fingerprint that best defines both these co-infections. And using these two unique fingerprints, we can then develop a rapid test for early TB detection. So what's worse than people dying from these diseases is the fact that we know it's treatable. And with high infection rates, Manitoba has one of the highest chances of people dying from both these infections across Canada. Early diagnosis is the key to managing and controlling TB disease. And so for my research, I aim to provide a rapid and affordable TB test so that anyone faced with these infections can get access to early treatments and get to live a complete and fulfilling life. And it all starts with finding these fingerprints. Thank you.
All right, thank you very much, Erwin. So next up, our second challenger is Alexander Arudjo, who is from the Natural Resources Institute. Alexander's thesis title is Impacts of Restoration, Assessing if Grazing by Reintroduced Plains Bison Affects Bird Abundance and Diversity in Banff National Park.
In 2017, Parks Canada placed 17 plains bison into the backcountry of Banff National Park. This is a hugely significant project for the species because most plains bison herds that exist today live on small patches of land that can only support small numbers of animals. The Banff herd, by contrast, is able to support as many as 1,000 animals over a 1,200 square kilometer reintroduction zone. This is a really cool project because every day these animals are out there, we're learning more and more, not only about how they live in a mountain environment, but how they transform it. See, bison are what we call ecosystem engineers. So they modify the habitats they live within through activities like grazing. And understanding how they do this is really useful, not only for land managers in Banff National Park, but for anyone who wants to put bison back onto their landscape. Think about it. If you're a land manager who has, say, an endangered species living on your landscape, you don't want to put that species at further risk by reintroducing the plains bison. So we want to learn from these animals, but the Banff herd provides a kind of an interesting challenge right now. We only have 62 animals, and they've only been there for 5 years. Now, you're probably thinking that's a pretty far cry from the 1,000 or so I just described. Well, we can look at some of the early effects right now to help give us some clues as to how they're transforming the land. Specifically, what I'm interested in looking at are birds. See, birds are very sensitive to changes in their ecosystem. Specifically, they're sensitive to changes in vegetation structure. Think things like the height of a plant. So for example, we have species X that likes really tall grass and species Y that likes much shorter grass. If you put bison onto a landscape, they will preferentially graze certain patches, right? They're not going to uniformly go across the land. So you'll have areas on the landscape where you'll have really short grass that species Y likes and ungrazed areas that species X really likes. My project involves going across the reintroduction zone and setting up monitoring stations where we can tell year per year how the bird populations are changing in terms of diversity, so the number of species there, and abundance, the number of members of each of those species. Then we go in and we look at how tall the grass is basically in each of those different areas. By comparing that to the data from the GPS collars the bison wear, as well as the number of dung pats on the ground, we can tell, okay, so if the bison are spending this much time in grazing this area at this level, we see this kind of change in the plant community, and we can link that to how the bird community is shifting in response. By creating these processes, these links, we can help create some information within 5 years of the project that's not only going to help out Banff managers again, but to help other land managers in the future who are going to continue to reintroduce bison across North America, ensuring that we're going to have these animals for centuries to come in a way that's healthy for them without putting other species on the landscape at risk. Thank you.
Okay, thank you very much, Alexander. So our third challenger today is Behzad Fakour from the Department of Mechanical Engineering. Behzad's thesis title is The In-Space Propulsion System for an End-of-Life Deorbiting Module for Small Satellites, or SCOT.
Okay, my project is about a prevention method to bring back CubeSats from the Earth. When humankind enters to a new place, it usually destroys totally. They destroyed Earth, and now it's turn for the space. My project is about to address this problem. Right now, I want to talk about space debris. They are numerous, they are hard to detect, and more importantly, they are dangerous. They have the speed of a bullet, and they are very, very fast, so they can make any disasters in the space. And for example, in 2020, ISS has to move and repositions 30 times. I want to give you an example. In a space, debris with the size of 10 centimeter can destroy the whole ISS in just within just seconds. So it is a very important thing that we are going to address that. And according to mitigation standard practices, but the US government and from the United Nations, we need to do three things. We need to prevent by design, capture it, and remove them. And my project, Scott, is trying to work on the prevent area. We are going to work on a propulsion system to work on satellites, CubeSats, small CubeSats, to bring them back to the Earth and have them burnt up after their mission. So it's kind of atmospheric re-entry, but there are other methods moving to in other orbits or direct retrieval. We know ourselves as space warriors who try to protect the space and bring the life back to the humankind, the life that they deserve. Thank you.
Okay, thank you very much, Behzad. So up next is Christophe Turcotte van der Rijt from the Department of Biological Sciences. Christophe's thesis title is Investigating the Plasticity of Migration Timing to Environmental Variability in Purple Martins.
You have probably heard about climate change, warmer springs, and how migratory birds arrive at our latitudes earlier each year. But have you ever thought about how these migratory birds seems to always know when there is an early or late spring, even though some of them travel tens of thousands of kilometers to get here? This is exactly what I want to address by looking at how purple martins adjust their migration strategies. But first, I want to stress how important it is for nesting birds to match their timing with their environment. Indeed, for the survival of the nestlings, hatching dates must coincide with the peak of insect availability, which occurs earlier in warmer springs. Thankfully, birds can remedy to this by changing their migration timing. This means that the same bird might arrive earlier at its nesting site at two very different dates on following years. For my study, I look at temperature, wind, precipitation, and day length. Day length being the length of light hours during the day. This can limit the time available for birds to forage or to migrate. The objective of my study is to investigate how these environmental factors might influence migration speed, stopovers, which is how martins will stop to recuperate along their journey, and the timing of spring arrival. To do so, I'm using the exact migratory route of over 300 individual purple martin, which had been tracked using very small, tiny backpacks. The daily location of each bird is associated with each of my environmental factors and is summarized for each migration. My predictions were that with few hours of day length and with poor weather like rain, cold temperatures, or opposing winds, these would reduce the migration rate and increase the number of stops as it would become more physically costly to migrate. However, my preliminary results showed quite the opposite trend. Except precipitation, which still slowed migration. These results indicated that cold migration tracks with short day lengths seem to be associated with fast migrating martins, which stopped very little. This could simply indicate that in favorable conditions, martins just spend more time foraging. But really, these unexpected results indicates that migratory birds are still hiding some of their secrets. Once I finalize my results, my research will provide guidance to which extent migratory birds can adjust to a changing environment. This will undoubtedly allow us to better preserve them.
Great, thank you, Christoph. So challenger number 5 now is Marissa Jampolsky from the Department of Interior Design. Her thesis title is Zakor, a Holocaust Memorial Museum for Mourning and Healing.
Hi, my name is Marissa Jampolsky. I'm a Master of Interior Design candidate in the Faculty of Architecture. Throughout my life, there has been an overarching theme of anxiety over the retention of history and identity. This anxiety is not related to my individual identity. Instead, it is of my collective identity as a Jewish person. Never forget was a phrase constantly recited to me surrounding Holocaust remembrance, and I sought to do just that. This discourse has led me down a path of research, a quest to absorb all the media I could concerning the Holocaust. This ranged from watching films and reading books to visiting historic sites and Holocaust museums. I put in the work to remember the events and understand them as if memories of my own. Upon entering the academic world of architecture and interior design, I set out to explore how space can aid in the quest to never forget. My thesis, Zachor, concretizes Jewish mourning ritual into a museum environment to aid in healing, mourning, and remembrance practices surrounding the Holocaust. Zachor seeks to develop a format for meaningful exhibition space that not only catalogs information, but also elicits a physical, emotional response from visitors, tying the present-day body to the memories of the past. This will be achieved through the exploration of theories, including Juhani Palausma's phenomenology of space and memory, as well as Alison Landsberg's research on experiencing another memory as one's own, known as prosthetic memory. The combination of these and other memory-based theorists are used to prove the critical connection between interactions with space and memory production, as I develop an interactive exhibition space to maintain modest and localized Holocaust remembrance. Judaism's mourning ritual also plays an integral role in not only maintaining memory, but also in assuaging the anxieties many feel surrounding the loss of eyewitness testimony. These rituals aid the mourner in their healing while also concretizing the memory of the deceased within their community. The Holocaust removed much of the opportunity to properly remember and mourn individual victims. Through embedding these rituals into a physical space, the visitor will be able to mourn and remember the victims in a religiously motivated and psychologically beneficial environment that is proven to aid in the core functions of memorial. Beyond memory and religion, the space also integrates the adaptive reuse of the Vancouver Unitarian Church. It is critical to the project to select an existing space, and I have utilized Graham Brooker's theories on reprogramming to ensure the building's past, present, and future are honored. We live in a world filled with contempt toward one another, but I believe much of this to be a misunderstanding. By producing a space that personifies the effects of hatred on individuals, I hope to open opportunity for further acceptance, conversation, and coexistence. Thank you.
All right, thank you, Marissa. So up next is Challenger 6, followed by a brief 3-minute break to allow the judges to finalize their notes and comments on the scorecards so they get a chance to catch up with all of this great work. So our 6th challenger is Olya Mihalituk. She's in the Department of Pharmacology and Therapeutics, and Olya's thesis title is Shining Light on Microglia Responses in Alzheimer's Disease.
One Sunday morning, I came to visit my grandparents. I saw my grandma, who recently retired, and she asked me what day it is today. I said, "It's Sunday, Grams." During my visit, she asked me that same question multiple times. Over the years, she started having difficulties preparing meals, paying her bills, and relied on my grandpa for most of her daily activities. She was diagnosed with Alzheimer's disease. It was heartbreaking to watch my grandmother, once a successful dentist, progressively lose the ability to carry out simplest tasks. You might already know that the main driver of Alzheimer's progression is accumulation of amyloid beta. These misfolded proteins are sticky and they clamp together. They block the signaling and therefore communication between the brain cells that are called neurons. Destructive processes start in the region of the brain called hippocampus that is responsible for forming memories. That is why memory loss is the first symptom. As disease progresses, different regions of the brain are affected, which leads to drastic mood changes, increasing confusion, and difficulty sleeping. But what if I tell you that the changes in the brain start more than 20 years before people get their symptoms? And by figuring out what is the driving cause of the progression of Alzheimer's could make us stop or prevent the onset of this disease. Research shows that inflammation is the key player in Alzheimer's. But what is the cause, and what is the meaning, and why is this the focus of my research? To consider the meaning of inflammatory response in Alzheimer's, we have to understand what function our innate immune response has. It is protecting us against different pathogens like bacteria, fungi, or viruses. And the cells in the brain that represent innate immune cells are called microglia. Microglia are like the bodyguards in our brain. They sense a foreign substance and elicit immune response, which includes releasing inflammatory factors and clearing the pathogen, which in our cases, amyloid beta. However, if microglia cannot clear the pathogen, it will have neurotoxic functions and inflammation won't resolve. Microglia will stop doing a lot of beneficial things for our brain and will start interfering with neuronal communication, contributing to memory loss. Work in my lab shows that by manipulating microglia responses and preventing prolonged brain inflammation, we can possibly preserve neuronal function and prevent memory loss. In my project, I'm looking at the strengths of neuronal connections by measuring electrical activity between the brain cells. I inhibit microglia responses and see what effect it has on memory in mice models of Alzheimer's. I see that even 2 hours exposure of amyloid beta can decrease memory. Imagine what a lifetime can do. Even though we're still gaining insight on the role of inflammation in Alzheimer's, it is worth doing it. And I think, and I hope that through this work, we will be able to help someone else's grandma. Thank you.
All right, thank you, Olya. So we'll now take a short break to allow the judges to total their scores so far and make notes on their score sheets. It's also time for viewers to take a stretch and replenish your snacks, and we'll return in 3 minutes. All right, welcome back. So let's continue with the competition and with challenger number 7. That's Matthew Cook, who is in the Department of Psychology. His thesis title is Computational History, Using Semantic Models to Measure Changes in Attitudes, Values, and Beliefs from Language.
The language that we use reveals a lot about ourselves and the way that our minds work. So it reveals our personalities, our mental health, and even our attitudes and our beliefs. For my PhD thesis, I wanted to study changes in language over time to see how changes in language reveal changes in our attitudes, our values, and beliefs. So I collected a huge body of digitized newspapers to analyze that spanned over 200 years from the late 1700s up to the present day. The problem was, however, with billions of words of newspapers, I had more newspapers than I could ever expect to read in my lifetime, not to mention it before I ever hoped to finish my degree. So to solve this problem, I had machine learning models do the reading for me. Now, machine learning models are a type of artificial intelligence that learn from nuanced patterns in data. And I programmed this model to read through this large body of newspaper data so that I could use the model's knowledge of language to inform my analysis. However, as you know, computers are not very good with words or language, but they are really good with numbers. So I programmed the computer model to represent each word as a long list of numbers. And importantly, similar words like dog and cat have similar numeric representations or lists of numbers compared to unrelated words. So what this let me do essentially is mathematics with words. So I used a formula to compare words to each other to see which words were related or unrelated to each other in expected or unexpected ways. And I measured how the meaning of words changed over time. So this allowed me to measure changes in our attitudes towards events like war, to track stereotypical beliefs towards social groups, and even to identify historical events like previous pandemics. I also did a bunch of other really interesting analyses, but I'll have to wait for a 6-minute thesis competition to tell you about those. Now, this work is important because the language processing methods that I'm using and adapting and inventing allow for the accurate modeling of language to understand how our attitudes, values, and beliefs are changing over time. The methods I'm using are also completely generalizable, so they work with any language data. So next I'm going to apply these analyses to analyze social media data, like from Twitter. My thesis is also important because it shows the potential of incorporating artificial intelligence in our research by using both human intuition and judgment to determine problems that are important to study in the first place, and by incorporating the raw power of artificial intelligence to analyze data and detect patterns in truly massive data sets. I believe that by taking this balanced approach, we can solve problems that can't be solved with more traditional research methods. Thank you.
All right, thank you, Matthew. So our next challenger is Shana Giesbrecht from the Department of Microbiology. Shana's thesis title is Molecular Detection and Quantification of the Prevalence of Sexually Transmitted Bloodborne Infections in Canadian Wastewater Samples.
A major concern we have all had during this pandemic is that COVID would find its way into our community undetected. Given the limited accessibility of clinical testing, how would our community leaders even know there was an outbreak until it was too late? Well, that's where my project comes in. My project focuses on using wastewater, also known as raw sewage, for tracking infectious disease. So how on earth can you track infectious disease using sewage? Well, for certain infections, people shed viral or bacterial particles in their urine or stool, and those particles end up in community sewage. We can isolate those particles and extract their genetic material in order to determine if a particular infection was present or not, and if it was, how much was present. Because sewage is communal, you get unbiased data with coverage from the entire community. How? Well, I think author Taro Gomi said it best in his 1977 children's story. Everybody poops. During the COVID-19 pandemic, many remote communities here in Canada began using wastewater testing to monitor for COVID-19. There are actually a few examples of communities who found COVID during wastewater testing when no known case existed within the community. This prompted them to implement widespread asymptomatic testing, and they were able to successfully identify the asymptomatic individual. After the success of using this strategy of surveillance for COVID-19, some communities expressed interest in using this strategy for other infections, including sexually transmitted infections. My job is to develop methods for the detection of common STIs in wastewater, including gonorrhea and syphilis. In Canada, the rates of gonorrhea and syphilis have gone up by 400 to 500% in the last 10 years, and STI cases are known to be greatly underreported, so improved surveillance is drastically needed to slow what can only be described as an epidemic. The hope is that wastewater testing can help us identify regions with high rates of STIs so that public health can allocate resources to affected communities. This includes healthcare and education. This is especially impactful in Northern Canada, where the rates of common STIs are 10 to 20 times the national average, and access to healthcare can be incredibly limited. Overall, targeted allocation of healthcare resources to affected communities represents an equitable allocation of healthcare resources for all. I think that this project has the potential to make a real impact, so thank you for taking the time to listen to me today.
Great, thank you, Shayna. Challenger number 9 is Pranav Mishra from the Department of Pharmacology and Therapeutics. Pranav's thesis title is Potential of Sex Hormone Estrogen in Prevention of Alzheimer's Disease, a Sex-Based Study.
What if I tell you I forgot what I was going to say? Fortunately, that's not the case today. We all have moments every now and then when we forget things, and yes, it doesn't seem to be so serious. But this lady in the bottom of the picture, she went out to walk her dog and came back alone. And let me tell you, she did not lose the dog. Rather, she completely forgot she even had one. Isn't this forgetfulness serious? Such loss of memory can worsen to a point where people have forgotten who they are. So when they went out, they never came back as they couldn't find their way back home. Sadly, in Canada alone, over 500,000 people are suffering from such loss of memory. And this condition is called as Alzheimer's disease. It's a neurodegenerative disorder characterized by increasingly worsening memory with age. And sadly, 65% of those suffering from it are women. A key fact that most of us know is that with aging, females undergo menopause. But what you probably did not know was that with menopause comes major changes in the body. One such change is a rapid decline in the levels of sex hormone called as estrogen. This hormone is a special chemical that plays an important role in memory formation in the brain and is the key focus of my PhD thesis. Falling levels of estrogen can lead to increase in inflammation and decrease in mitochondrial function in the brain. Inflammation, a process that is there to protect the brain, turns against it and now eats it up. Parts of the brain literally break down over time, while mitochondria, which we all call the powerhouse of the cell and has the potential to generate energy, it also stops functioning properly. Remember how electrical appliances get damaged with power fluctuations? Similarly, the brain gets damaged with mitochondrial dysfunction, and all of this contributes to loss of memory. And even though majority of people suffering from it are males, the rest are females. So in my study, I'm going to focus on both males and females at the same time, and I'm going to make use of not one, but 2 animal models for a more reliable outcome. Although Alzheimer's was first recognized as a disease over 100 years ago, we still don't have an effective treatment. However, I strongly believe I'm on my way to find one. As in my experiments, I've been able to increase the energy levels in these brain cells using estrogen. And yes, the results, they are promising. This disease alone demands a lot of attention globally because sooner or later, it's going to affect most of our lives as we are all associated with the aging population, including our own parents. But let me tell you, there's nothing to worry about because estrogen alone has the potential to keep the memories safe. And soon, anyone who goes out, they will be able to find their way back home. Thank you.
Great, thank you, Pranav. So next is Stephanie Borles from the Department of Biochemistry and Medical Genetics. Stephanie's thesis title is Exploring New Targeted Therapies for Sonic Hedgehog Medulloblastoma.
We all live in fear of the dreaded C word. Yes, I'm talking about cancer. It is now estimated that 50% of us will be diagnosed with cancer in our lifetime. That means that every second person you know will be diagnosed with some form of cancer during their lifetime. Now, picture this: a young child. They're innocent, carefree, laughing, adventurous, full of energy, unaware of the harsh realities of life. This precious human being has just been diagnosed with brain cancer. A young child that has only just begun their life has already been diagnosed with a life-altering and life-threatening disease. Brain tumors are among the most prevalent forms of childhood cancers and account for 20% of all pediatric cancer diagnoses. Medulloblastoma is the most common pediatric brain tumor, and the average age of diagnosis for this tumor is between 5 and 9 years of age. Medulloblastoma is a very aggressive brain tumor, and the current treatment consists of aggressive surgery, high doses of chemotherapy, and radiation to the entire brain and spinal cord. And despite some improved clinical outcomes, 40% of patients still succumb to their disease, while survivors are left with extensive cognitive and physical delays following their treatment. I want to change this and help make a better life for these children. What if there was a treatment that could specifically target medulloblastoma tumor cells and kill them without harming any other cells in your body? There are currently 4 different types of medulloblastoma, and my research focuses on the type known as sonic hedgehog. My thesis is investigating targeted therapies to treat sonic hedgehog medulloblastoma that have the potential to reduce toxicity, improve quality of life, and increase survival for these young patients. I recently discovered that a drug named trametinib can kill sonic hedgehog medulloblastoma tumor cells, and that this treatment significantly extended survival in mice with sonic hedgehog medulloblastoma. My goal is ultimately to identify drugs that can be translated into early clinical trial development to generate new and innovative targeted therapies so that the next time a child is diagnosed with medulloblastoma, their future and survival will not be questioned. John F. Kennedy once said, "Children are the world's most valuable resources and its best hope for the future. We need to protect them and ensure their survival." Thank you.
All right, thank you, Stephanie. So our 11th and final challenger is Akshay Malik from the Department of Physiology and Pathophysiology. Akshay's thesis title is Doxorubicin-Induced Heart Failure, the Role of Endoplasmic Reticulum Stress.
Let me tell you a story of almost over 50% cancer survivors. However, it is a heartbreaking story. It starts with the discovery of an anti-cancer drug, doxorubicin, which has saved millions of lives by killing cancer cells. But soon after, we realize that it loves oxygen a little too much. And you tend to stick around someone you love, right? So where can doxorubicin find most oxygen in the human body? The heart. Doxorubicin molecule can utilize oxygen to stay active, and in the process, it generates reactive oxygen species, which can kill a heart cell. Even though heart cells have defense mechanism to fight back, doxorubicin kills defense as well. Well, heart cells are irreplaceable. Once dead, they do not regenerate. So what would researchers do? Well, they also looked into the heart cell, and they looked where they could find most oxygen, in the powerhouse of cell, mitochondria. Sadly, after 50 years of research and over 10,000 research papers, we still do not fully understand how in just 24 hours, doxorubicin can kill 60% of heart cells in a culture dish. Sad, right? But I won't be here telling you this story if it was the end. Well, as a researcher myself, I also looked into the heart cell, and a part of cell which is responsible for the contraction and relaxation of heart muscle was all stressed out. I am talking about protein factory of the cell, endoplasmic reticulum. Doxorubicin treatment can impair the structure and function of endoplasmic reticulum to an extent that the cell suicide is the only option left. Well, after this finding, I did not stop. It was time to give heart cell its defense mechanism back by using a molecule called interleukin 10, which is an anti-inflammation molecule, but it can also maintain oxygen balance both in the mitochondria and endoplasmic reticulum without interfering with the anti-cancer property of doxorubicin. I found that the interleukin 10 treatment prior to doxorubicin can also foster good structure and function, as well as promote cell survival to up to 90% even after the doxorubicin treatment. Well, my research is a step forward towards interleukin 10 therapy in conjunction with the doxorubicin so that we give our cancer survivors a better lifestyle. And you do not have to listen to any more heartbreaking stories on this stage or elsewhere. Thank you.
Great, thank you, Akshi. So this completes the Challenger presentations for this afternoon. Thank you to all of the challengers who presented today. All of them were outstanding presentations on a diverse set of research topics. And I'd like to give you a virtual round of applause. I wish we were all in the same room so we could give you a real one. The judges will now meet virtually in a breakout room to total their score sheets and determine the winners. The judges' results will be announced live in just a few minutes, so please don't go anywhere. While we wait for the judges' results to come back, there'll be a short slideshow introducing you to some of the university's past 3MT winners. And so I'll invite you to enjoy the video, and we'll be back shortly. So, welcome back, everybody. The judges have made their decision. I want to express that it was not an easy task because all of the challenges were outstanding. And they did want to mention that they were very impressed with the content of the presentations, the presenters themselves, and especially all of the hubbub involved with living in an online world and presenting in an online format rather than in a real life scenario. So in no particular order, the challengers who will advance to the finals on April 7th and receive a $200 prize are Pranav Mishra, Alexander Aruju, and Ulya Mihalichuk. So congratulations to all of the winners. There will be a wild card selection announced after the final 3MT heat. The wild card will be selected by a committee who will be attending all 3 of the 3MT heats and voting on their choice from all of the competitors from the 3 heats. The wild card selection will also advance to the finals, but will not be awarded the $200. The wild card selection will be notified by email early next week after all 3 heats are completed. And it's been a wonderful day of diverse ideas and exciting research. Thanks to all of the challengers for sharing your research with us. I would like to thank our technical team of David and Teddy, who were instrumental in facilitating the live event today. Thank you to the students, their advisors, and their friends and family for all of the support that they've given. And finally, I also want to thank our terrific judges, Mr. Greg Mackling, Dr. Sandra Weber, and Dr. Jake Stout. So before we break, I want to give one final reminder that the University of Manitoba 3MT final competition is online on April 7th at 7 p.m. Everyone is welcome to attend. The link will be on the U of M Faculty of Graduate Studies webpage at umanitoba.ca/3MT. Thank you all for attending wherever you might be today. And on behalf of the Faculty of Graduate Studies, I wish you all a great afternoon.